Lyme arthritis is a very severe, debilitating symptom associated with Lyme disease, and it affects a large number of individuals. According to the CDC, approximately 300,000 cases annually of Lyme disease are reported to them. I am very interested in understanding the wide range of clinical presentation of Lyme disease. You can take 10 individuals and they're all infected with the bacterium that causes Lyme disease, and some individuals on one end of the spectrum will be essentially asymptomatic, have very little, if any, response to that bacterium that causes the disease. On the other end of the spectrum, you have individuals that are suffering from very severe, debilitating arthritis. In terms of the nature of the arthritis that develops in the mouse joint versus in the human joint, they are remarkably similar. A lot of the same cells that are recruited out of our bloodstream to kill bacteria when we're infected with Borrelia burgdorferi, those same cell types in the mouse are recruited into their joints to try and kill the bacteria. We identified a specific protein on the surface of these cells that are responsible for killing and clearing the bacteria. In the absence of this specific protein, the severity and the duration of inflammation is much worse. So we can take a mouse that normally has a very mild, if any, inflammatory response to Borrelia, and if that mouse is deficient for this one specific protein, they have much more severe, debilitating, persistent inflammation. How does this play out in the human Lyme arthritis patient population? We're now working very closely with collaborators at Massachusetts General Hospital, physicians that actually see these patients, these Lyme arthritis patients that are refractory to antibiotic treatment and have more severe disease. And we are pinpointing the same molecular regulation of severity and persistence of inflammation in the human patient population as we observe in the mouse model of Lyme arthritis. And this is an absolutely critical connection between the basic immunological research that's done here and the more patient-oriented translational research that's being done at some of the top hospitals in the country. Inflammation is the way in which our immune system fights bacterial and viral infections. So if we had some magic pill that would completely block inflammatory responses in our bodies, I think we'd have a much, much shorter lifespan, because the bacteria, the viruses, the fungi that infect our bodies would be able to cause damage that could not be controlled by our immune system. So we don't want to completely eliminate inflammation. On the other end of the spectrum, we don't want inflammation to run rampant, to be so severe, so strong in response, of such a long duration, that it causes collateral damage to other tissues. So I have this overarching Goldilocks hypothesis that we don't want too little inflammation, we want just the right amount. We don't want too much inflammation, because in addition to killing the bacteria, that's going to cause collateral damage. We want just the right amount. And individuals being individuals, some people have too exuberant a response to the bacteria, so we have to help them temper that inflammatory response. With other individuals that might suffer prolonged bacterial infection because they don't have enough of an inflammatory response, maybe we want to immunoengineer them to elicit a little more of an inflammatory response than they normally would. Conservatively, less than 5 years away. I think we are really close to uncovering that fine balance